All right, let's dive right into this explainer and decode some of the true microbiological mysteries of the Bacillus genus. If you're currently preparing for your veterinary microbiology exams, or maybe you're just getting ready for clinical practice, you are absolutely in the right place. We're going to break down everything you need to know about these highly resilient, environmentally persistent pathogens, and we're going to extract just the highest yield facts directly from the source material so you're totally prepared.
But hey, before we get completely bogged down in the pure microbiology of it all, I want you to ponder a bit of a clinical mystery. What exactly connects a 50-year-old pasture, a sudden bovine death, and a case of human food poisoning? I know, it sounds a bit like a riddle, but it perfectly illustrates the environmental persistence and the incredibly diverse impacts of Bacillus bacteria across the entire One Health spectrum. Keep this riddle in the back of your mind as we unravel how one single bacterial genus can be responsible for that whole shebang.
Okay, so here is our high-yield clinical blueprint for today's explainer. We'll start with Section 1, Meet the Bacillus family. 2, Anthrax, Pathogenesis and Disease. 3, Clinical Diagnosis and Action. 4, Outbreak Control and Treatment. 5, the One Health Picture. And finally, Section 6, a high-yield exam review to test your knowledge. Let's get right to it. Section 1, Meet the Bacillus family. Large, gram-positive endospore-producing rods. Right off the bat, the crucial point here is how we visually and biochemically differentiate Bacillus. These are large gram-positive rods. And when I say large, I mean they can reach up to 10 micrometers in length, which is pretty massive for a bacterium. They form highly resistant endospores, occurring singly, in pairs, or in these long chains. But here's your first really high-yield exam fact. They are aerobic or facultatively anaerobic, and they are catalase positive. This is absolutely critical because it perfectly distinguishes the Bacillus species from the Clostridia group. Clostridia are also gram-positive endospore-forming rods, but they are strictly anaerobic and catalase negative. It's also worth noting that the vast majority of the 50-plus Bacillus species out there, they're simply motile saprophytes. They just hang out in the environment, doing their thing with zero pathogenic potential. But the ones that are pathogenic, well, they are incredibly important. Now, this is where things get really interesting, especially for your exams. There's a strict dichotomy between the two major species you will definitely be tested on, Bacillus anthracis and Bacillus cereus. If you're looking at them on a lab bench, their colonies can actually look kind of similar. They're large, maybe with a greenish tinge for B. cereus, or that classic Medusa head appearance for B. anthracis. But paying close attention to their clinical differentiators is basically a cheat code for microbiology. B. anthracis is non-motile, non-hemolytic, and totally susceptible to penicillin. B. cereus, it's the exact opposite. It's motile, hemolytic, and resistant to penicillin. Plus, B. cereus has strong, rapid lysinase activity on egg yolk agar, whereas B. anthracis is notably weak and slow. Moving into Section 2, Anthrax, Pathogenesis and Disease, Unlocking the Deadly Mechanism. Okay, so the virulence of B. anthracis relies on two main things, both encoded by plasmids. First, a capsule made of poly-D-glutamic acid that inhibits phagocytosis. And second, a highly complex toxin. This toxin mechanism literally operates like a Trojan horse. It has three components that, believe it or not, are completely non-toxic on their own. Step 1, protective antigen, or PA, acts as the binding moiety. Think of it as the master key to the host cell. Once PA binds, it unlocks the cell, allowing the other two factors to sneak inside. Step 2, edema factor, or EF, enters. This is an adenylate cyclase that massively increases cyclic AMP levels. That causes the severe fluid accumulation and tissue swelling we see clinically, and it also shuts down neutrophils. Step 3, lethal factor, a zinc metalloprotease, gets in and stimulates macrophages to release massive amounts of cytokines, things like TNF-alpha and interleukin-1 beta. This targeted cellular payload results in severe, rapid shock and death. But how on earth does a herd get exposed to this in the first place? Well, it is absolutely vital to understand that environmental persistence through sporulation is the single most critical factor in the epidemiology and spread of anthrax. These spores can literally lay dormant in the soil for decades. We're talking 50 years or more. They are unbelievably resistant to desiccation and high temperatures. Outbreaks often happen when something disturbs the soil, like heavy flooding, excavation, or even earthworms bringing these buried, decades-old spores right back up to the pasture surface where susceptible herbivores just unknowingly ingest them. Section 3, Clinical Diagnosis in Action, Safely Identifying the Threat. Picture this scenario. You arrive at a pasture. You find a bloated bovine carcass. You notice there's incomplete rigor mortis, and you might even see dark, unclotted blood issuing from the nostrils or anus. This is the absolute classic presentation of percute septicemic anthrax. So, as the clinician on site, here is the most important question you must ask yourself. How exactly do you go about necropsying this animal? You don't. Seriously, you absolutely do not. This right here is the ultimate clinical rule of negative. You must never, ever open a carcass if you suspect anthrax. Inside that dead animal, the bacteria are just existing in their vegetative state. If you cut into that carcass, you instantly expose those vegetative cells to ambient oxygen. That oxygen triggers massive immediate sporulation, and you have literally just condemned that pasture to decades of severe environmental contamination. So if you can't open it, what do? Instead of a necropsy, you carefully follow these exact diagnostic steps to protect yourself and the environment. In ruminants, you'll just draw peripheral blood from the tail vein using a sterile syringe and make sure you apply 70% alcohol-soaked cotton to the site to prevent any leakage whatsoever. If it's a pig, you'd collect peritoneal fluid. Then, you make a thin smear and stain it with polychrome methylene blue. When you look under the microscope, you are hunting specifically for square-ended, blue-staining rods surrounded by very distinct pink capsules. Remember, that capsule actually diminishes over time after death, so fresh smears are totally key. Oh, and a quick tip for your exams. While you might hear about the Ascoli thermoprecipitation test, it actually lacks specificity because B. anthracis shares thermostable antigens with other completely harmless Bacillus species. Just stick to the morphological and biochemical profiles. Section 4. Outbreak control and treatment. Containing the spores. Once a diagnosis is confirmed, it is all hands on deck. The crucial takeaway here is that managing an anthrax outbreak requires a massive, multi-pronged approach. First things first, if you catch it early, in-contact animals can actually be treated with high doses of penicillin G or oxytetracycline. You also must immediately report suspected cases to your local regulatory authorities. In endemic regions, you'll be using the Stern strain live spore vaccine. This uses spores that essentially convert into non-encapsulated, avirulent organisms to build immunity without causing the disease. On the strict biosecurity front, you need 5% formalin or 3% peracetic acid foot baths at the entrances. Any contaminated buildings require really intense fumigation with formaldehyde. And finally, what do we do with those unopened carcasses? They have to be incinerated or buried very deeply, far, far away from any watercourses, and scavengers must be kept strictly away. Section 5. The One Health Picture. From microbe to ecosystem. Let's zoom out a bit and look at the broader One Health ecosystem. This table perfectly captures the One Health relevance of the entire genus. The Anthracis is obviously the heavy hitter, right? It causes fatal septicemia in ruminants, localized edematous swelling in pigs and horses, and cutaneous, pulmonary, or intestinal anthrax in humans. But look at the others. Bacillus cereus is a massive issue in human food safety, known for causing severe food poisoning, though it can occasionally cause rare mastitis in cattle. And then there's a really interesting emerging pathogen, Bacillus licheniformis. Generally, this is just an environmental organism associated with food spoilage, but in veterinary medicine, it's increasingly linked to sporadic abortions in cattle and sheep, and this is specifically tied to the feeding of moldy hay or silage. All right, Section 6, High-Yield Exam Review. Let's test your knowledge. Here is case number 1. A cow is found dead on a pasture, blood is issuing from the nostrils, and there is no rigor mortis. Based on everything we just talked about, what is your immediate diagnostic action? Think back to our strict field diagnosis rules. What do? If you were practically screaming, "Do not open the carcass" at your screen, you nailed it. You absolutely do not open that carcass. You safely collect tail vein blood using a sterile syringe. If you had decided to cut into that cow, you'd have exposed vegetative bacteria to oxygen and just earned that pasture a permanent spot on a 50-year biological hazard list. Next one, lab analysis. You isolate a large, gram-positive rod that is motile, hemolytic, and penicillin-resistant. Is this Bacillus anthracis? This tests your ability to recall those strict differentiators we established earlier. No way! This biochemical profile screams Bacillus cereus. Remember, B. anthracis is notably non-motile, non-hemolytic, and penicillin susceptible. B. cereus and B. anthracis might be closely related endospore-forming aerobes, but these specific opposite biochemical differences are literally your key to an accurate, life-saving diagnosis. Okay, let's test your molecular memory. Mechanism. In the pathogenesis of anthrax, what is the specific role of the protective antigen, or PA? Let's see if you can recall the sequence from our Trojan horse narrative. Protective antigen acts as the critical binding moiety. Remember, PA doesn't do the cellular damage itself. That is the job of the edema factor and the lethal factor. But PA is the master key. It binds to the host cell and unlocks it, allowing that lethal payload to get inside, disrupt water homeostasis, and trigger a massive cytokine release. Well, you've done an awesome job navigating the high-yield realities of the Bacillus genus today. From basic gram staining rules to managing a full-scale farm outbreak, you now have the clinical blueprint. But considering that B. Anthracis spores can survive in the dirt for 50 years or more, just waiting for the perfect moment to germinate, it begs the question, will our agricultural environment ever be truly free of this microscopic threat? We'll leave you with that provocative thought to ponder, just to remind you that in veterinary microbiology, the past can literally rise to the surface at any moment. Keep studying, stay curious, and we'll see you in the next explainer.